Right, everyone, just welcome back. Right, welcome to our third tutorial on vision. Okay. So to start things off, I just want to hear from all of you, just to do a quick revision. Colors of the rainbow, right? Which color has short wavelength? Which color has long wavelength? Yeah. I'll stop for a while. I'll listen to the answers. Let's continue. All right. So everyone, the answer just to rephrase myself, short wavelength is blue or violet. Then the long is red or yellow, right? So on this slide, the eye, this is practically speaking just a revision of yesterday's lecture. I think I talked about the cornea. Yes, I did. Right? I talked about anterior chamber. I talked about the pupil. Right? The lens, vitreous chamber. I talked about all these things. Right? Retina. Right? Yeah. So for landmarks of the retina, we talk about all these regions. Right? So we have this optic disc. Right? The so-called blind spot. Right? So the question is really answered for you. And then we have macular, right? The fovea. Basically, the fovea contains all our cones, right? And the periphery of the fovea, just for everybody's knowledge, right? It really contains a high density of rods, yeah. So not cones, we're about rods. Just take note of that. So the optic disc is actually special. It doesn't really contain any photoreceptors because it is a spot, right? Okay, the spherical space that actually sends out all the optic nerves to our brain, right? So cannot really have any other interference from any other cells, right? It's just an opening to our brain. So just take note of it. No photoreceptors. And this is just a sagittal view, right, on the right panel, right, from the side, a lateral view from the side here. Demonstrating the blind spot. Okay, this is from our textbook. I'm not too sure how many of you people can see it, right? So, but try. Okay, close your left eye, focus on the dot. If you have your textbook, that would be great, right? It should be done in a more natural fashion. Okay. Yeah. So eventually, you should move your head forward, right? Come close to the image until the stack of money disappears. Okay, try that. We see. All right. Transduction of rod photoreceptors. This is just a revision from yesterday's lecture. Very important, by the way. I want everybody to get this. So a simple question. So when the light photon, when the photon reaches a photoreceptor, what happens to the photoreceptor? Is it depolarized or hyperpolarized? Right. Note the actions of the sodium ion channels. Right. Okay. So a question is why. There is this important why concerning the rod photoreceptors. So I just read out everything. So why are photoreceptors active in the dark and less active in the light, and not the other way around? Yeah. So this is an answer from the conservation of energy perspective. Yeah, I really think this is one of the best answers. This is not just the only answer. Just take note, right? So because majority of the time, right, we are under daylight. We're working under daylight. So it is just much more efficient, right? Physically speaking, physiologically speaking, for us to, activate the release of certain neurotransmitters, amino neurotransmitters like glutamate, for instance, right? For the least amount of time. We don't want to keep on releasing them. So as said to the other students, it's better that we kind of reuptake all these amino acids, and convert them into proteins. We need proteins to make cells, right? To make neurons, to make muscles. Imagine if you keep on releasing them into the synapses, ultimately they'll be broken down. Right, they'll be broken down, they will be urinated away, no good, right? So activating photoreceptors in the dark, so they will become less active under light conditions, right? So as a way to conserve energy. Everybody got this? Right? Very, very logical, extremely rational. All right, so everyone, so concerning this slide, what I said just now, from our textbook, what I personally do find confusing, I still do find it confusing, is this panel here, right? Okay. So it's showing the photoreceptors on top instead of the bipolar cells on top, right? But do know that the photoreceptors belong to the innermost layer of our retina, right? It shouldn't be placed at top, And then look at this horizontal cell, right? So it's so important. The horizontal cells always gives you an idea of where the light is coming from. So its soma is always pointing towards the source of the light, meaning that the light is coming in the bottom-up direction. It's coming from below rather than on top, right? and this makes it very non-intuitive. And this, this bolded line here, so it's supposed to give you a cross-sectional overview of the photoreceptors. But at the same time, it also shows you the direction of lateral inhibition, right? That really runs in a parallel direction, right? In the horizontal direction. Later we'll get to that, right? And consequently, right, I prepared this image, okay, which is a 180 rotation, right, as mentioned just now, right? So this is ganglion cell on top, okay, and then photoreceptors below, right? So light is coming from the top, okay, reaching our ganglion cells, as I mentioned, right? Ganglion cells send all this information all the way down to the photoreceptors. Photoreceptors will process the color, right, and non-color information, motion, contrast, and then reprocess all this information, send it outwards back to the ganglion cells. Ganglion cells then send them to the brain. That's how it's like. So it's a U-shaped loop. And then receptive field, as what I mentioned, take note, right? Primarily it's the photoreceptors, right? The photoreceptors make up the receptive field. Not made explicit by our author, very unfortunate, right? An activity of a particular visual interneuron. So if you do not really read into the text, you can easily mistook, right? You can easily mistake this visual interneuron to be something that's other than the bipolar and ganglion cell. So I do not want you guys to think in that way, right? So we're talking about bipolar and ganglion cells as recipients of all the information from the photoreceptors, right? Yeah. Which type of cell is this? Again, this is from our textbook, right? The same image again, made smaller. So we're looking at an on-center off-surround cell, right? So the on-center is always depolarizing, always, right? And then its periphery, its immediate periphery is hyperpolarizing. So this is what this second slide is showing you, right? When the light falls in the surrounding photoreceptors, right, the cell hyperpolarizes. The cell in question, the cell, singular term, right, we're actually talking about this bipolar cell, right? Take note of that, right? Because the horizontal cell is concatenating, it's combining all the inputs from the rods, the cones, and then sending them to one bipolar cell in this figure. And this is what I mentioned yesterday in lecture as well. So what happens if the same amount of light, right, falls in the surround and the center of an on-center of surround cell? What happens is that we won't get any change in depolarization, no change in neural activity, right? So because excitatory response and inhibitory response, right, from the center, the surrounding, respectively, they cancel each other out. Yeah. All right, and that brings us to lateral inhibition. It is a mechanism, right? It is a mechanism, the neural process or procedure that maintains this center surround organization, right? Always remember this thing. Always tie it back to the center surround organization, right? So this is directly from the textbook. Okay, I paraphrase it a little bit, but just take note of the words in bold. So the first one is compete. So what cells are competing with each other? Right? Essentially, it's like photoreceptors and horizontal cells serving the on-center, right? So the ones that's being marked in gray in our previous slide, right? The gray cells, photoreceptors, they compete with each other. They're competing with all the cells in the lighter areas, right? To activate the bipolar cell, right? Which is receiving all the inputs, right? All the information, right? From the photoreceptors. And the direction is horizontal, right? In the direction running lateral or parallel to the surface of the retina, right? As shown by the bolded line in our previous slides, right? So, and then the second one is essentially it's a kind of elaboration, right? So active photoreceptors, horizontal cells will limit, right? The keyword here is limit. It will constrain the activity of neighboring less active cells, right? So you have to mention these two concepts, right? And then this is what will give us, allow us, right, to see changes in light and dark area, to see the boundaries of bars of color, right? And then you will allow us to see increases and decreases in contrast, right? Later on, we have a video to show that, and this is a video. Okay, we'll watch the video. All right, so everyone, this slide and the next one were from my older collection of slides back in the States, right? So I just want to show you, right, all this concept, right? Remember, it's ganglion cell we just watched in the video, not bipolar cell, right? So for ganglion cells, we're talking about spiking potential, right? Action potentials. Right? So note that this is what is being shown. For half the video, we're looking at this on-center off surround ganglion cell, right? So the plus basically just refers to depolarization, right? The presence of light shining on it, and then we have depolarization. Right? If it's in the off surround, and when the light is shining on it, we don't really get as many action potentials. And the reverse happens, right, from the off-center on surround ganglion cell. So literally the light, in order to produce depolarization, it has to occur in the surround, right, the spot in the surround. Then we get lots of action potentials. In the center, no, right, negative, there are hardly any action potential on this slide. So this is really for everybody's general information, retinal information processing, right? So we had this person, okay, I think he's German, right? German scientist Koffler, he mapped out the receptive fields of individual retinal ganglion cells in the cat, right? That's what I mentioned. And he found out that the spatial layout of the ganglion cell receptive field is concentric. Wow, okay. Similarly for the bipolar cell, right? The circle is always the best shape. And the cell fires at its fastest rate when the spot size matches the size of the excitatory center, reduces its firing rate when the spot begins to encroach on its inhibitory surround. That's for the on-center ganglion cell. Right? The off-center cell does exactly the opposite. So refer back to the previous slide. So what happens after all the information reach our ganglion cells? And how does all this information end up in the brain? Right, so this point in time, we have to talk about the events that happen after the optic nerves, right, leave our retina, right? So from the retina to the brain, this is also a revision, right, from one of our slides from yesterday's lecture. So 1, 2, and 3, right? 1 is the optic chiasm, right, where the optic nerves converging point close to the anterior commissure. Number 2 is the lateral geniculate body, right, the LGN, lateral geniculate nucleus in our thalamus. And of course, the last part is our visual cortex. All right, impossible to get it wrong. And this is a very nice diagram, again, from our textbook. It's a little bit small, right? But I just want to point out, okay, the numbers, very, very small, I know. Very hard to see. You've got to zoom it up on your own computer, right? So basically, 1, 2, 3, 4, right? This is, if you rotate the head around, this is actually the person's right visual field, right? And this 6, 7, 8, and 9 belongs to her left visual field, right? So both our eyes, can be separated into both left and right visual field. And if you zoom up the diagram, right, you can see, I'm just looking at the right eye. So from, let's say from 9 all the way until 6, the nerve fibers from there. So they're colored in red. Yeah, they're colored in red. So just follow my cursor. And they're going like all the way to the left hemisphere, the left visual cortex. So, but take note, it's only like the images, the optic nerves, right? The optic nerves is receiving inputs from the left visual field of the right eye. Okay, what happens to all the optic nerves on the right visual field of the right eye? They do not automatically go to the left hemisphere. They are ipsilateral. So in this case, they are going to the right hemisphere. So take note of that, right? Right. So the axonal outputs, I would say, the axonal outputs to the visual cortex, where they land ultimately, it really depends on which visual field of the eye in particular, or which eye are you talking about, right? So similarly, the logic of what I just mentioned is actually a little bit different from the left eye. So when you look at the left eye, what happens, when you look at the left visual field of the left eye, the red neural fibers, they do not cross over directly to the right hemisphere, the right visual cortex. They're going to the same side, they're ipsilateral, right? So whatever is on the left visual field of the left eye goes to the left striate cortex, okay, or visual cortex here. And whatever is on the right visual field of the left eye, ultimately they cross over. This one they cross over to the other side. All right, they cross over to the right visual cortex. So something to take note of, okay? This is very complicated, okay? I will not test you people to label the visual fields and to color the neural fibers. That is really too much, okay? If you're doing neurosurgery, then this is a testable, this is like a must, all right, for the neurosurgeons to label, right? But for us, no. All right, so it's quiz time again. Let's do this test. All right, everyone, thank you for the discussion. So on this slide, okay, sensation, visual sensation to perception. This is again a revision, quick revision from yesterday. So we had two streams that's found in the 80s, right? It's still here, but we still have them. One is the dorsal stream, the occipital-parietal stream, and then the other one is the ventral stream, right? Which is also called the occipital-temporal stream, right? So the dorsal stream just basically tells us where something is located. That's why it's called a where stream. And then the ventral stream is essentially called the what stream, right? Giving us the identity of objects of where we are, right? Yeah. Or what we're doing at the particular position, I would say that, to be more precise. So we'll watch a short video later, right? So this is really about monocular versus binocular cues. I want people to kind of take note of the binocular cues, just for the sake of general knowledge. Really, really important. So this panel is essentially just showing you the presence of retina disparity. All right, when we have both our eyes open, in certain spots, coordinates, depending on where our head is oriented, will appear closer to us than other coordinates. But it's just that the way our retina works, right? It really integrates, compresses all this information. So everything is subconscious now, right? And then our brain actually could not really tell all these different coordinates apart. But essentially, we're actually seeing different coordinates. Yeah. All right, let's go and watch the video. All right, vision loss. So on this slide, I just want to mention the most common cases of visual deficiencies, right? So this is myopia, nearsightedness, versus hyperopia, farsightedness, right? So it's basically myopia is just caused by elongation of eyeball, right? So our eyeball is not really perfectly spherical, right? And then for people with myopia like myself, right? So it's kind of elongated, unfortunately. So the focal point is actually changed, right? So basically the acuity, right? The concentration of all the light rays, they do not fall neatly, do not fall accurately on the fovea, right? So, unfortunately, many objects to us will seem fuzzy, right? So we need to get a concave lens, right, to correct for that. So for farsightedness, they use convex lens. Doesn't look nice, right? Because you really want to stretch out the light wave, right? So the focal point actually will reach the person, will reach the fovea much earlier than a person with perfect eyesight. So in that case, this is actually, you don't really get an elongation of the eyeball, but your eyeball is actually kind of squeezed, it's a little bit squeezed along the horizontal axis, yeah. So it's a bit kind of oval shape in a vertical direction in this case. And then interestingly, some people with myopia, they can't get hyperopia in old age, right? How is that even possible? That's possible when you have something like some loss of vitreous humor, right? Vitreous humor that just leads to your eyeball changing its shape again, right, along the vertical axis. So it is possible, right? And it is a blessing in disguise. Okay, so another short revision, right? Rods and cones. Which one is responsible for night blindness? Okay, we know that it's rods, right? Always rods. Yeah. Color blindness will be cones, right? And then scotopic versus photopic vision. Scotopic would be related to our night vision, so it's related to rods, right? Photopic, as you can see from the word photo itself, right? Related to color vision, right? Related to daytime, right? And vision loss. So this is just to give an idea, not just our eye, not just our retina. Damages to our optic nerves, our LGN, thalamus, optic tract radiation, all the white matter fibers, right? Plus primary occipital lobe, right? And of course, the brain regions lying along our dorsal and ventral streams that will lead to vision loss, right? Because we'll be losing all these cortical neurons, right, responsible for processing visual information. So lastly, we'll watch this very interesting video, right? This person has visual agnosia, right? Let's just watch this. All right, thanks everyone. So that was a amazing video. I really recommend everybody to rewatch it when you have the time. So this is our last activity, mix and match for today, right? This is really for everybody's general knowledge, education, not examinable. But I do think that this is important, right? This is really to help us become more educated.